Have you ever really stopped to think about how a person, well, begins? It all kicks off with this absolutely incredible microscopic journey that plays out in just 2 weeks. This is the story of how you were first sketched out, literally the biological blueprint for life. So let's dive in and see how it all starts. It all comes down to this one single, incredibly dramatic moment. A sperm cell meets an egg. And look, this isn't just some casual meeting. This is the spark. This is the trigger that ignites an extraordinary and, you'll see, perfectly timed chain of events. And that's the big question, isn't it? I mean, how on earth does one fertilized cell transform into something with a defined top and a bottom, all in just 14 days? Well, the answer lies in a perfectly orchestrated, incredibly precise timeline. Okay, so our story officially begins at day 0. This is the moment of conception, what we call fertilization. You can think of this as the starting pistol for the entire race of development. So what is fertilization exactly? Well, it's the fusion of two highly specialized cells, the sperm and the egg, to create a brand new single cell called a zygote. And this one cell contains the complete genetic instruction manual for a totally new individual. And this massive event doesn't just happen randomly. Nope. It takes place in a very specific spot, the ampulla, which is the widest part of the uterine tube. And it's from here that this brand new zygote is going to start its long journey. This whole moment is actually a multi-step invasion. It's pretty wild. First, the sperm has to get through the egg's outer layer of cells. Then, it pulls off something called the acrosome reaction, where it releases these powerful enzymes from its tip to basically digest a path right through the main protective shell, the zona pellucida. Once it's through, their membranes fuse, and boom, the genetic material from both parents combine to form a completely new and unique nucleus. So the zygote is formed, and right away, the clock is ticking. This first week is just an intense, non-stop journey of division and transformation, all while it's traveling toward its final destination in the uterus. The pace of change here is just staggering. By day 1, we're at 2 cells. Day 2, 4 cells. By day 3, it's this solid little ball of over 16 cells called a morula. Then by day 5, it develops a cavity, becoming a blastocyst. And finally, around day 6, it starts to hatch. It's just a relentless forward march of progress. Now, this rapid-fire multiplication is called cleavage, and it's got this one really weird, really cool feature. The cells keep dividing and getting smaller, but the whole thing, the entire embryo, doesn't get any bigger. It's actually a really clever biological trick. You get a ton more cells really fast, but you don't need a huge amount of energy for overall growth. It's all happening inside that protective shell, the zona pellucida. Right around the morula stage, the very first big decision gets made. The cells literally sort themselves into 2 teams. The ones on the outside, they're destined to become the placenta, the life support system. And that little group of cells on the inside, well, that's the part that's going to become the embryo itself. That's the future you. Then, as fluid starts to seep in, that solid ball of cells rearranges itself and it forms this hollow structure we call the blastocyst. You can see the key parts here. You've got the inner cell mass all clustered at one end, the outer layer called the trophoblast, and this new fluid-filled cavity in the middle. But before it can do anything else, the blastocyst has to pull off a great escape. It has to break free or hatch from that protective zona pellucida. It's kind of like a chick hatching from an eggshell. And this is an absolutely critical do-or-die moment. If it can't pull off this escape, it can't implant in the uterus, and the whole journey just ends right here. All right, welcome to week 2. So our little blastocyst has made its journey and successfully hatched. Now it's facing its next huge challenge, implanting into the uterine wall and, well, setting down some roots. So here's the crucial thing to understand. The blastocyst doesn't just gently land. No, it actively invades the nutrient-rich lining of the uterus. This aggressive process, called implantation, is all driven by some very specialized cells that are about to get to work. And here they are, our implantation specialists. That outer cell layer splits its work into 2 distinct teams. First, you've got the cytotrophoblast. Think of it as a cellular factory, on the inside, just churning out new cells. Those new cells then merge together to become the syncytotrophoblast, this powerful, invasive outer layer that actively burrows into the uterine wall to lock everything into place. Now, I know what you're thinking. The second week sounds incredibly complicated with a lot happening all at once. But luckily, there's a really simple, really powerful way to remember it all. We call it the week of twos. The rule could not be simpler. Almost every single major development that happens during week 2 happens in pairs. It's a fantastic mental shortcut for getting a handle on this whole complex period. Let's just run through them real quick. The trophoblast, which we just saw, splits into 2 layers. The embryoblast, that inner cell mass, also forms 2 layers. 2 brand new cavities appear, and then finally another supportive tissue layer also splits into 2. See? Everything in 2s. And here is the first and, honestly, the most important 2 for the embryo itself. That inner cell mass flattens out and splits into 2 distinct sheets of cells, an upper layer called the epiblast and a lower layer called the hypoblast. Together, these 2 layers form what we call the bilaminar germ disk, and this is the fundamental foundation from which the entire body will eventually be built. So as week 2 starts to wrap up, all these pairs of structures have been formed. The embryo is successfully implanted, and then one final crucial event happens that really truly establishes the body's fundamental plan. Right around day 14, this small, localized thickening of cells appears. It's called the prechordal plate. Now, it might not look like much, but its significance is absolutely immense. This is the very first structure that gives the embryo a direction, an orientation, a top and a bottom. Just like the source material says, this is the first sign of cranial caudal orientation. For the very first time, this little flat disc has a designated head end and the tail end. Without this axis, there's no up or down, no front or back. This is the master coordinate system that every future structure, from your brain to your spinal cord, is going to be built on. The blueprint is officially set. So, there you have it. In just 14 days, we've gone from a single cell to a fully implanted, 2-layered disc that knows which way is up. The architectural plans are complete. The next big question, of course, is what happens when the real construction begins?